In this video, we are going to be talking about the concepts of chapter 14, which has to do with viruses. First, we're going to talk about what is the definition of a virus. A virus is a particle that contains a small amount of genetic material and it is surrounded by a protein envelope. Now, this genetic material that viruses have can be DNA or RNA. Understand that another word for virus is going to be virion. As you can see illustrated in the left side of the slide, we have an image of a virus or a virion. And in the bottom of it, we have different types of viruses. Even though that for this course, you are not entitled to know the different type of viruses, I wanted to illustrate how different they look like. Now, as you can see, at the center of the image that we have in the top left corner, we have the nucleic acid. And as I just mentioned, it can be DNA or RNA. Now, the nucleic acid is actually surrounded by capsid subunits known as capsomeres. And when those capsomeres come together, they make the capsid, which is the protein coat that some viruses have. Now, many viruses, in addition to that, have a membrane envelope. And at that membrane envelope, you can observe some spikes or projections that are actually protein derived, and understand that these spikes are going to help them to attach to the host cell. Viruses are organized by their genome and their existence of an envelope. As you can see summarized in table 14.4 that it can be found on the textbooks page 424, the table summarizes 4 different classes of viruses overall. As you can see, pretty much what we have is DNA containing that are enveloped, DNA containing that are not enveloped, RNA containing that are non-enveloped, and RNA containing that are enveloped. Understand that for the exam, I will be asking questions pertaining this slide specifically, in which it can be classification of a particular virus, or if you're given the name of a virus, what type of disease actually is created by it. Now, the reason why viruses are not considered organisms is because they cannot survive for long periods outside a host cell. Okay, so since they always have this dependence on the host cell, that's why we consider them particles and not really organisms. Viruses have a specific life cycle, and as you can see, it is illustrated in the figure on the left side of the slide. These are the different steps for a virus life cycle. First, the virus is going to bind the cell. I'm going to highlight the steps as we go. So step number 1, we have the virus binding to the cell and is going to release its nucleic acid into the cell. Understand that in the illustration that I have here specifically, this is going to be a DNA containing virus. On step 2, this nucleic acid, and again, as I mentioned in this illustration, it is DNA, is then replicated and transcribed by the host polymerases. As you can see in the replication, we're making more of that DNA product. And in the transcription, as you can see, that allows that RNA to then be translated to create the proteins that are important for building those viral particles again. So on step 3, as you can see, we have specifically how the transcription and translation are going to be building those coproteins of the capsid. As you can see on step 4, we have the assembly. And on step 5, specifically, during the lytic pathway, is going to have those viral particles lysing the host cell. In addition to the lytic cycle, viruses can also undergo a lysogenic cycle. Okay? In a lysogenic cycle, basically what we're going to have is that the viral DNA is going to be incorporated into the host chromosome. So as you can see illustrated on the image on the left side, these are the steps for the lysogenic cycle. Okay? First, as you can see, we have a prokaryotic cell on number 1. Then we have the particular virus, in this case is a bacteriophage, that is going to attach and release its DNA, shown in red, into the prokaryotic cell. As you can see on illustration number 3, we have that phage DNA that is going to move through the cell to the host DNA. On step 4, we see that phage DNA, meaning in this case the virus DNA, integrating itself to the host DNA and is creating what is called a prophage. As you can see on number 5, this prophage then remains dormant until the host cell divides. And understand that as a whole, once the cell undergoes cell division and gets divided, then understand that that phage DNA that was brought in by the bacteriophage, in other words, the virus, is going to begin express itself. Okay, some of the cells are that contain the prophage go and create new viruses or phages, which then will go and infect other cells. So the thing is that when we compare these two pathways, it is important to note that the differences between them is that in the lysogenic cycle, the spread of viral DNA occurs through a usual prokaryotic reproduction. Okay, so what happens is that, remember that once that DNA from the bacteriophage is incorporated into your prokaryote, that it's going to be dormant, and through cell division then it can lead to activation. Now, when we have the lytic cycle, understand that the results of incorporating the DNA from the virus into our specimen, which in this case we're just illustrating it through a prokaryote, is going to be more immediate because the copies of the virus are going to be created very quickly and the cell is going to be destroyed. Now, When it comes to the viruses, and remember, I just mentioned in a few moments ago that we have viruses that are going to be DNA containing viruses and other ones that have RNA. Well, viruses that have RNA, not all of them, but a number of them are considered retroviruses. And retrovirus specifically are going to be viruses that take their RNA molecule and they convert it to DNA. Okay? So, understand that in the image that we have on the left side, it actually summarizes how that viral RNA gets transformed into DNA. As you can see, the viral RNA is released into the host cell. That's going to be the first step. Then the virus itself has an enzyme that is called reverse transcriptase that is going to synthesize double-stranded DNA from that initial viral RNA that was placed by the virus particle. Then that double-stranded DNA is incorporated into the host DNA via recombination using these sequences that are called long terminal repeats or LTR sequences. After that, the DNA is going to be transcribed to RNA, which is then packaged into new virus particles. Understand that one of the systems that is well understood that is a retroviruses is HIV, which is the causative agent of the acquired immunodeficiency syndrome, in other words, AIDS. Next, we are going to see how HIV viruses actually infect human cells. And as you can see, it is illustrated in the following steps. On step number 1, and I'm just going to highlight as I go. As you can see, HIV is going to fuse to the host cell surface. And as you can see, the specific protein that is identifying, because remember that this happens to be in the immune system. So it's actually T cells that get affected by these viruses. So the immune cell is what gets identified by the virus identifying this protein that is called CD4. Then this retrovirus, as you can see, HIV RNA, reverse transcriptase, integrase, and other viral proteins are going to be entering the host cell. And as you can see, everything is being delivered at this moment. Then, as you can see, the viral DNA is formed. Let me highlight step 2. The viral DNA is formed by reverse transcription. Remember, the reverse transcriptase is the enzyme that takes that RNA back to double-stranded DNA in the host cell. Then that viral DNA is transported across the nucleus and it integrates into the host cell. As you can see, integrase is going to be specifically the protein that is going to allow that to happen. Once that viral DNA it is integrated into the host cell, as you can see, that is called as a provirus molecule. Then that host DNA that contains the viral DNA that was brought in by the integrase and it was incorporated into the genome, is going to be transcribed, is going to be translated. So that's why step 5 illustrates that that new viral RNA is used as genomic RNA to make viral proteins. Then, as you can see on step 6, the new viral RNA and proteins are going to move to the cell surface, okay? And a new immature HIV forms. And lastly, the last step in the life cycle of HIV, as you can see, the virus matures when proteases release the protein that form this mature HIV. And as you can see, it creates a mature virus particle or virion.